In the previous several lectures, we focused on the great variety of living things, as well as the shared characteristics of cells and the molecules and the genetic machinery of all life. We're now going to shift our focus to the origin of life. The question is, how did life arise? For thousands of years, humans have pondered this deep mystery which lies at the heart of philosophy and religion and, of course, science. The question in detail is likely to remain unanswered for many, many decades, but the question is framed by one of the great ideas of biology. That is that life arose from air, water, and rock by chemical reactions. In Lecture 54, we're going to consider the chemical steps by which non-life became life. After a somewhat philosophical introduction, we're going to focus on three aspects, three historical stages of this scientific process. First, I want to look at Louis Pasteur's sound refutation of the spontaneous generation hypothesis. Then I want to look at the original and clever experiments of Stanley Miller and Harold Urey. And finally, I'm going to talk about the discovery of a deep, hot biosphere and a new theory about the origin of life. Let's step back from the scientific research and consider some of the philosophical aspects of this question of life's origin. Is it appropriate for science to even ask a question about life's origin? And I think the answer is maybe yes, but maybe no. It really depends. There are three possible scenarios for life's origin. The first is that life may have been a miracle. It may have been a divine act of intervention. It may have been that God said, "Let there be life," and there was life. If so, then the origin of life is not a scientific question. There's no experiment you can propose. There's no observation you can make to study a miracle. A miracle is by definition outside the known natural laws. So if that's the origin of life, it's really beyond science. It's also possible that the origin of life was an event that's fully consistent with the known laws of physics and chemistry, but an extremely improbable, perhaps a unique event, perhaps an event that only took place on Earth. And once again, if this is so, if the origin of life was a unique event, it's really not amenable to scientific study, because you can't go into the laboratory and study a unique event. But there is a third possibility, and that's that life is an inevitable consequence of chemistry. That given an appropriate environment, an appropriate planet with water, for example, sufficient time, that life always arises. And it's only this third option, that life is inevitable, or at least highly probable, that presents the prospect of scientific study. If life is likely to arise whenever and wherever appropriate conditions occur, then scientists can study life's origin in a laboratory through experiments. You can actually duplicate those conditions, you can study chemical reactions, perhaps eventually even create primitive life forms in the laboratory itself. Scientists learn about the origin of life in a couple of different ways. They observe life on Earth today. They see the characteristics of living things, from extremely primitive single-celled organisms to very complex multicellular plants and animals. They also have an extensive fossil record, which we'll look at in a couple of lectures. Fossils that tell us about the progress, the development of life over time. This epic history stretches back almost 4 billion years of Earth history. That history of life can be conveniently divided into two parts, and this defines a two-part scientific strategy for studying life's origins and development. The first period is called the era of chemical evolution. That covers the time from the Earth's first formation up to the appearance of the first object we could call a living thing. Then, once life appeared, comes a period of biological evolution. That's when organisms began to compete with each other for a limited amount of resources. And so you have biological evolution in the process of life changing over time, which is going to be the subject of the next 3 lectures. It's very difficult to know what that first life form was like. And it may have been vastly different from any sort of life that's on Earth today. For example, it probably was not exactly like a prokaryotic cell that we see today. It was not like a simple microbe. It's very likely the very first life forms didn't have DNA or RNA to pass on information. It may have had a much simpler means of reproduction and passing on information. Perhaps it didn't even have proteins or enzymes in the way we think of them today. The first life form, for example, might have been an extremely thin two-dimensional surface coating on rocks. That's a real possibility. Something that's just a few molecules thick. This would be an invisible coating. You wouldn't even be able to see it if you held that rock in your hand. It would just sort of gradually spread and expand over mineral grains. But whatever that first life form looked like, it certainly had to possess three key characteristics, characteristics that are shared by all living things today. And let me review those three characteristics. First of all life obtains energy and raw materials from its surroundings. It has to have atoms, it has to have energy, and it probably was carbon-based. In fact, it almost assuredly was carbon-based, since all life forms today are carbon-based, and carbon is the only element we know of that's versatile enough to produce a whole range of molecules that might lead to that first life form. Secondly, all life has the ability to grow. And surely whether this original life form was two-dimensional or three-dimensional, whatever it is, it had to grow also, because it had to go from the first point of life to expanding and fill the entire globe with life. And finally, all life has the ability to reproduce with variations. Now, you can imagine a two-dimensional surface reproducing by growing and expanding, and then perhaps a rock splitting in half, or through some sort of earthquake, you actually could divide a surface into two surfaces and then keep fragmenting and splitting apart. That's a possible way of growing. You also could have variation in a two-dimensional form, because it might have, for example, a layer of organic molecules that were protecting it, a kind of membrane. But remember, there's such a variety of carbon-based molecules that membrane could vary from place to place in the exact kind of molecule that was included. And over time, the membranes that were most effective at keeping the outside out and the insides in might have been selectively taken, and so you might have had evolution in that sort of way. So life may have been very different, but it had to contain those three properties. Well, let's look now at the history of this great question of the origin of life. The history of these origins places our current work in a context. 2 centuries ago, ideas about origin of life were very different, because there was an ideal championed by Aristotle called vitalism, and that idea was still very much in vogue. According to this doctrine of vitalism, a life force permeates the cosmos, and as a result, life arises spontaneously all around us, all the time. The question of life's origin, therefore, wasn't even asked in the modern sense. Think about your own experience, and you'll see why this idea of spontaneous generation of life is not such a crazy idea. You observe, for example, if you leave a piece of bread lying out on a counter, mold just spontaneously appears on it. If you leave meat out on a summer day, eventually maggots are just going to appear. They seem to rise right out of the meat. You see new growth in the spring. In winter, life seems to have died around us, and yet new plants constantly rise in the spring. So the idea of spontaneous generation is not such a strange one. But this idea was soundly trounced by Louis Pasteur, the great French chemist, who was instrumental in abolishing these ideas and leading us to a new way of thinking about life's origins. Pasteur was born in 1822. He was the descendant of generations of tanners, and so had a great deal of practical chemical experience in his family's background. As a boy, he was only interested in drawing and thought he wanted to become an artist, but he excelled at school as well, and eventually he attended university in Paris. He focused on physics and chemistry, and then throughout his life he ascended to higher and higher positions of honor in the French scientific establishment. Eventually he became the founding director of the Pasteur Institute, which was an institute founded in his name and in his honor. He was so revered in France at that point. Pasteur made major contributions to crystallography, to the understanding of fermentation, which was, of course, a subject of great interest to the French wine industry and the beer industry there. But his major discovery, the thing that really established his fame in France, was the discovery of the germ theory of disease. In other words, he's the one that discovered that microbes cause infectious diseases. And of course, once you find the cause of a disease, you're much closer to finding a cure. He suffered a stroke at the age of 48, but in spite of the fact that it partially paralyzed him, he remained dynamic and incredibly active and productive until his death in 1895 at the age of 72. It's no doubt that his reputation as a scientist was enhanced by an exceptional ability at public speaking. He was a great debater. He also was able to focus his keen scientific intellect on problems of great social concern. So he was one of those rare scientists who saw his science in the context of a much larger world, and that also gained him a great opportunity to make contributions to his native France and to the world in general. Okay, back to the origin of life now. Pasteur did not accept the idea of spontaneous generation, and he conducted a brilliant series of experiments to show that water, when it was boiled, could be sterilized. What he did is he sealed some beakers, and he kept some open to the air, and then he boiled the water. What he found is that when you boiled the water and sealed the container, they became sterilized or pasteurized. They would not allow other life to form. And yet, if you kept the beaker open, then eventually microbes would get in and start contaminating that water. These ideas had tremendous practical consequences. His development of pasteurization transformed the production and preservation of food. For example, it revolutionized the distribution of milk and of beer at that time in the 19th century, and really changed the whole way food is transported and stored. In the process, Pasteur also introduced the dictum that no life can occur without prior life. Think about that. No life without prior life. This brilliant discovery has the consequence that life's origin is pushed way back to an inconceivably remote time and place. How could anyone make observations or experiments to study such a remote event? And so the origin of life almost became a subject beyond science because it was pushed back to a time when no one could study it. Pasteur's ideas did generate a few intriguing suggestions. Charles Darwin, for example, he's the great British scientist who we're going to learn a lot more about in a couple of lectures. He developed the theory of evolution by natural selection. He wrote the following quote to a colleague in a letter. Quote, "If we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc., present, that a protein compound was chemically formed, ready to undergo still more complex changes." Here Darwin realized that life's origin, however it occurred, depends on three key resources. First, life requires water. That's Darwin's warm little pond. Second, life requires energy. Perhaps the sun, perhaps it was lightning, or maybe it's the Earth's inner heat, as we'll see. That's a possibility too. And then life requires chemicals. Carbon, oxygen, hydrogen, sulfur, perhaps with some nitrogen and phosphorus thrown in. This is so you can make those molecules of life. Remember the lipids, the carbohydrates, proteins, nucleic acids. You need all those basic building blocks. Early in this century, in the 20th century, several scientists expanded on these ideas that were contained in Darwin's letter. Most notable amongst these researchers was the Russian chemist Alexander Oparin, who lived from 1894 to 1980. In 1922, while he was still in his 20s, Oparin proposed the idea that life arose from a body of water that gradually became enriched in organic molecules. This is the so-called Oparin ocean, or sometimes known as the primordial soup. You just gradually built up supplies of organic molecules dissolved in the ocean. Somehow, he argued, these molecules became self-organized into a chemical system that could duplicate itself, and that was the first life form. Now, these are intriguing ideas, and they're certainly subject to testing in the lab. But it wasn't until those exuberant years following World War II that a systematic experimental approach to the study of this primordial soup was devised and implemented. Okay, we have the requirements of water, of energy, of chemicals, and this actually points to an experimental strategy, a very simple strategy for studying the origin of life. The first of these experiments were devised and conducted in the early 1950s at the University of Chicago. They were done by chemist Harold Urey and his graduate student Stanley Miller. They're often called the Miller-Urey experiments. Miller and Urey started with glassware, actually a device not unlike Pasteur's experiment. In this case, there's a sealed flask. It's partially filled with water, and that represents a primitive ocean. Now, the rest of the flask is filled with a simple mixture of gases to represent an early atmosphere. They used methane, CH4, ammonia, that's NH3, and carbon monoxide, some hydrogen thrown in, and then the water is heated. That gives you a warm little pond, if you will. And then they set off electric sparks, or they exposed their chemicals to ultraviolet radiation to represent that early atmosphere and the dynamic, energy-rich environment of the early atmosphere. This is a simple desktop experiment, and it represents the early ocean and the early atmosphere. At first, they started off with clear water, but within a couple of days, that water became cloudy, and then it started turning shades of brown and pink, indicating new molecules, new chemicals are being produced in that water. When Miller and Urey analyzed that water, they found a complex mixture of organic molecules. They found carbon-based molecules that are the building blocks of life. These were made simply from water and air with lightning or ultraviolet radiation to trigger the chemical reactions. Miller's first publication was a bombshell. He announced, quote, "the production of amino acids under possible primitive Earth conditions." This article appeared in the May 15th, 1953 issue of the prestigious journal Science, 1953, the same year that Watson and Crick discovered the double helix and announced that in Nature. Now, amino acids are only one of the building blocks, one of the key molecules of life. They're the building blocks of proteins, remember. When Miller's results first came out, the press had a field day. They said, well, we're just one step away from making life. They wrote about synthetic life crawling out of test tubes in the laboratory and so forth. Well, they were very far from the truth there. It made great hype, but in fact it took many, many years to understand the full significance of these Miller-Urey experiments. Thousands of subsequent experiments during the last 4 decades have defined the promise and also limitations of this idea that life arose at the surface of the Earth. Time after time, Miller-Urey experiments have demonstrated the synthesis of life's most basic building blocks. It's become like a cottage industry in chemistry departments. But as exciting and important as these results are, they're still daunting problems. First and foremost, the building blocks that Miller and Urey make are only the first step. They're only the single building blocks. You have to assemble them into polymers, into a large arrays. Living things are made up of much more complex groups of these smaller molecules. And the trouble is, these larger molecules just aren't stable in an environment of lightning and ultraviolet radiation. The polymers tend to break apart into the individual amino acids. You just can't form proteins easily, for example. So this leads us to a real major question. Where did life arise? If life can't possibly have arisen right at the surface of the ocean because the environment was too harsh, where else do you have? I want to tell you about an alternative hypothesis, one that's arisen in recent years to challenge the Miller and Urey model. The warm little pond, or the surface of the ocean, is not the only wet, energy-rich environment on Earth. We also have deep hydrothermal zones, especially places on the ocean floor near volcanic vents. Those are places where hot, mineral-rich waters flow through cracks in the rock. These become an active site, an alternate site for life's origins. Remember the spreading center, the mid-ocean ridges where new crust is formed in the model of plate tectonics? Those are active volcanic areas. Huge areas of the Earth's surface that are actively hot water, energy-rich zones where life might have arisen. There's several really good reasons to consider these hydrothermal origin of life hypotheses. The first and foremost are a dramatic series of discoveries that have been made in the last 20 years about deep life. Let's think about deep life in the ocean. We have to actually go back more than 120 years to the HMS Challenger expedition from 1872 to 1876. They conducted the first deep dredgings of the ocean, more than 100 dredgings bringing up life forms from more than a mile deep in the ocean, places where people thought that no life could possibly exist. Yet they discovered many strange new life forms, new species living in those black depths of the ocean. And then we fast forward 100 years to 1977, when Jack Corliss, an oceanographer at the University of Oregon, was engaged in a whole series of deep dives in a submersible called Alvin. He was going to these deep ocean ridges to study the ridge systems and their volcanoes. But he discovered an astonishing new series of communities, new ecosystems of life living right on those ridges, right where the volcanic waters were coming out, superheated waters in contact with deep, frigid, cold ocean water. An amazing new environment with rich life forms. These deep ecosystems are spectacular. New species of clams and crabs, shrimp, giant 6-foot-long tube worms that are blood red. You've probably seen pictures of these deep ecosystems, fascinating systems. But what may not be so obvious from the photographs is that these deep ecosystems are dominated by microbes, by single-celled organisms which are at the very base of the food chain. And these remarkable microbes are exceptional for at least 3 reasons. First of all, these microbes are largely independent of the surface. They don't use the sun's energy in any way, contrary to many textbooks that say the sun is the origin of all life. They rather obtain their energy by oxidizing or reducing minerals that spew out of the hydrothermal vents. So they gain energy through oxidation-reduction reactions, essentially using the chemical energy of the Earth, the chemical energy that's produced by the Earth's heat. These microbes have devised an astonishing variety of strategies. Up to 20 different kinds of redox couples are used by these microbes in order to gain energy. The second remarkable feature is these microbes require high pressure. For them, high pressures and high temperatures are not an extreme environment, they're the normal environment of life. And the third remarkable feature is that genetic studies show that these microbes are among the most primitive life forms on Earth. They're closer genetically to the oldest common ancestor than almost anything we see on the surface of the Earth. So these discoveries prompted Corliss and his co-workers to publish a remarkable and original idea that submarine hot springs might actually be the site of life's origins. Subsequent studies have shown that microbes thrive many places underground where there's rock and water. Indeed, if you drill a hole where you're standing right now, about a mile or 2 straight down, the chances are if you brought up that rock core and looked at it carefully, there would be life forms living in the rock. Any place where rock and water interact, there's likely to be microbes, because rocks turn out to be a good source of food for these microbes. Now, deep life is sparse, but the volume of the Earth's crust is immense, on the order of 1 billion cubic kilometers. So it's possible that the mass, the total mass of these tiny microbes is comparable to all the animals and plants, all the trees that you see on the Earth's surface. The Earth's biomass may indeed be dominated by microbes living on rock. There's some other reasons to consider this hydrothermal hypothesis, and these reasons come from geology. Recall that according to the nebular hypothesis that the sun, the Earth, and the other planets formed from a vast cloud of dust and gas, there was a great bombardment in which all these materials came together by gravity. And then you had volcanoes that formed the ocean and the atmospheres. Well, in the late stage of the Earth formation, any large impact would have come in, smashed the Earth's surface, and basically blown away the atmosphere, blown away the ocean, so that the entire surface of the Earth would have been sterilized, time and time again probably. However, if life arose in a deep hydrothermal zone, those microbes living in rock deep beneath the surface would be much more insulated from these insults. And therefore, it's very likely that deep life could have survived when surface life could not have. Another reason for studying the hydrothermal origin is from planetary science. And this is more a wish than a reason. It's because many planetary scientists have now realized that there are other hydrothermal zones on other planets and moons. Earth is the only known world with a surface ocean. But Mars must have had surface water early in its history, and it's very likely that Mars now has a subsurface water, subsurface hydrothermal systems that still exist. This means that if life was to arise at the interface between an ocean and an atmosphere, you're really restricted to the Earth and possibly Mars early in its history. If, however, you can originate life in a deep hydrothermal zone, then you can add the deep interior of Mars today, but also Europa, which is one of the moons of Jupiter, Callisto, another one of the moons of Jupiter, Titan, a moon of Saturn, and perhaps other bodies in the solar system as well, because there are many places where water is believed to exist in a liquid state beneath the surface of planets and moons. NASA is now planning missions to Europa, which is the most likely site for this sort of thing. Europa, it turns out, has a deep ocean beneath a thin crust, which is basically an ice surface crust over the entire moon. NASA is now displaying a lot of interest in deep ocean work research in Earth. And the reason is because if you can develop probes for the deep ocean on Earth, it's possible then you can send a spacecraft to Europa and probe Europa's deep ocean and therefore look for life and other geological type phenomenon in Europa. So that's why NASA is now supporting a great deal of oceanographic research. It seems odd that when we look up into the heavens, you actually can make your plans by going deep under the oceans of Earth. Well, let's think about scientific evidence for this hydrothermal origins hypothesis. If you're going to survive scientific scrutiny, you eventually have to do experiments, the chemical experiments, to show that life might have originated with rock, water, in a deep hydrothermal environment. This eventually can be bolstered then by experiments you do in the laboratory. It's really one of the wonderful aspects of science, that you can do experiments, that you can model the natural world in a laboratory environment and gain insight from it. This has become one of my real passions in scientific research right now. A few years ago, I got interested in this hydrothermal hypothesis, and at the geophysical laboratory where I do research, we began studying the deep hydrothermal origin of life. We conduct experiments in a very simple way. We take gold tubes. We put a little bit of rock, a little bit of water, a little bit of appropriate gas-type mixtures in these capsules, and then subject them to sustained high temperature and high pressure in an apparatus that squeezes and heats, much like a pressure cooker. We do this for controlled temperatures and pressures and times. We then open the capsules and we look and see what we've produced. Preliminary results show that you get extremely fast and facile reactions of organic molecules from just rock, water, and gas. In one set of experiments, for example, we put water, nitrogen gas, and powdered minerals. That's a good proxy for a primitive condition at one of these hydrothermal vents. And we produce huge amounts of ammonia. That's NH3. Now, ammonia is an essential starting material for amino acids and for producing other important biological molecules. So our work suggests that the hydrothermal vents was the principal source of the Earth's early ammonia. There are many, many other steps like this, and we're chemically, step at a time, we're going to study the whole process that may have led to the origin of life. Turns out this work may also solve another paradox, the early faint sun paradox. It turns out the sun early in its history was much fainter than it is now. And if you just take that radiant energy from the sun, the Earth should have been a frozen ball. Not enough sun energy was getting to it. But if you put a little bit of ammonia in the atmosphere, a very effective greenhouse gas, the Earth would have warmed up enough to make the oceans liquid. So our work is also pointing to a possible explanation of that paradox. In other experiments, we've synthesized amino acids, we've made lipid-type molecules, and this is ongoing research. We're very excited. It turns out lots of other laboratories are now joining in the fun, so you're likely to be reading about some of those results in your newspaper as well. Let me make a philosophical point about this. To some people, the warmth of the sun symbolizes life. It symbolizes heaven. And yet the idea of a sulfur-rich volcanic vent on the ocean floor, these hot sulfurous exhalations from the floor, would more symbolize hell, reminiscence of the old ideas of what hell must be like. How could life have arisen in a place that's so hostile, so unlike our benign surface of the world? It really goes against our sense of metaphor. Perhaps, though, the surface, which is bathed in ultraviolet light from the sun, where lightning is coming down, perhaps that really is on Earth the extreme environment. Perhaps we have to rechannel our thinking. Perhaps the deep hydrothermal zone is the most benign environment on Earth. Nature doesn't have to respect our metaphors, however cherished they may be. Well, to summarize this lecture, the modern subject of life's origins began with Louis Pasteur's refutation of spontaneous generation. Thus, rather than countless origin events that occur around us all the time, it turns out there was one origin event extremely distant in time and place. The Miller-Urey experiments demonstrated that biological molecules could be synthesized under conditions, controlled conditions representing the surface of the primitive Earth, an ocean and an atmosphere with lightning, with ultraviolet radiation. But those experiments have a problem because the ultraviolet radiation that catalyzes the reactions to make organic molecules also breaks apart proteins and other large molecules essential for life. Recent findings of a deep, hot biosphere have expanded the focus of origin research to hydrothermal zones on the ocean's floor, which may provide an alternative way of making these molecules. But however life arose, we can be pretty sure that once the first cell formed, life quickly spread around the globe and evolution began. And that's the subject of the next lecture.